Let's just go. So this is the Pathology Grand Round for all attendees. And it's a great pleasure to have Charlie Alpers give this talk on a real focus of his work over the last number of years. Just to be background, Charlie graduated from Yale, medical degree at University of Rochester, residency in AP, CP at University of Cal San Francisco, couple years research fellow in Brigham and Women's, and then hired at the University of Washington as a renal pathologist. And Charlie's done a number of things. The most memorable is probably going to be his work on toxin effect on fish in the Duwamish River, but we stray from the topic. So for everyone who has questions, please put them in the chat and then I'll read them. So Charlie, let's learn about diabetic nephropathy in models.
Well, thank you all for joining me. I'm going to use this time to try and compress about a 40-year experience with diabetic nephropathy into something less than an hour. And it's a bit of a challenge, but it's a good challenge. So let me begin with my disclosures. I have received funding support for the studies I'm going to share with you today, some from the NIH, some from corporate entities, but I will not be discussing how any of the therapeutics that I will discuss can be used for patient management. Three objectives here: understand the structural changes of diabetic nephropathy, understand the potential and barriers for regression of diabetic nephropathy, and understand how new therapeutics for diabetes may have benefits for regression of kidney pathology resulting from diabetes. So I'm actually going to just change one part of my screen here and go back. So diabetes is an enormous global problem, and this slide illustrates it from three perspectives. One is a global perspective, which looks at the world, and you can see the darker the color, the higher the prevalence of diabetes, and you can see virtually no region of the world apart from Greenland and Antarctica is spared from a growing prevalence of diabetic nephropathy. The middle graph is very disturbing. This involves a study in the United States, the Nutrition Health and Examination Estimation Survey, which looks over periods of time in the US at the prevalence of diabetes and its complication, chronic kidney disease. And you can see over an extended period of time, extending to the present, that the incidence and prevalence of diabetes continues to grow. Its complication, chronic kidney disease, as well as other complications, continues to grow. And that's in a period in which we've had a number of therapeutics, standard of care therapeutics, deployed with no real impact on this growing prevalence of chronic kidney disease. And we'll touch on that in just a short while. And the manifestations of chronic kidney disease that are emphasized in these studies are functional ones of protein excretion, damage to the kidney filter with loss of protein in the urine or albuminuria, reduced renal excretory function, considered as glomerular filtration rate, or a combination of the two. Apart from these renal manifestations, you can see that if you look at all causes of chronic kidney disease in the United States, using the most recent surveys, you see that diabetes by far is the largest etiologic cause of end-stage kidney disease in the United States, surpassing hypertension, glomerulonephritis, and other forms of kidney injury. So now I want to start the story of my personal engagement with diabetic nephropathy. And I'm going to take you back to the first year after which I graduated medical school. So in his brief summary of my career, let me lift off the fact that I first did an internship and residency in internal medicine. I did it at Boston University Hospital, and this is a picture of my residency class. And in case of you're looking for me in this group of hirsute men and some non-hirsute women, that's me in the front row in 1978. That was a particularly grueling residency, I would say even brutal, and has probably contributed significantly to the fact that I'm now a renal pathologist and not an internist. But there were some rewarding aspects of being a member of this house staff group, and some of you may recognize this individual over here. This is an earlier version of my wife, Ingrid, and we've now been married for nearly 40 years, so there were definitely some rewards to that internship and residency in Boston. But the real reason I wanted to show you this picture was to consider what was the therapy of diabetic complications in 1978, my internship year. At that time, we knew that the important thing to do was to control the patient's blood pressure. And actually controlling hyperglycemia, blood glucose, was controversial. At that time, there was a raging controversy that the better control of glucose would lead to reduction of diabetic complications, arguments led by investigators at the Joslin Clinic, while an equally articulate group of investigators at UCSF made the case that if you had good control of hyperglycemia, you did so at the cost of having multiple episodes of hypoglycemia in the patients with mini strokes and brain anoxia, and that this complication far outweighed the benefits of glycemic control. And it wasn't until 1993, with the publication of the Diabetes Control and Complications Trial, that the issue was settled, that the benefits of controlling hyperglycemia far outweighed the disadvantages of occasional episodes of hypoglycemia in patients. Treatment with renin-angiotensin system was just then being considered. The investigational drug at that time was called captopril, and it was being studied as an investigational drug at BU at the time I was an intern, but only for patients with heart failure. And the nature of the study was such that patients with diabetes were specifically excluded from the trial. So these are treatment with drugs that are either an angiotensin converting enzyme inhibitor, such as lisinopril, something we now pay a lot of attention to because of the role of the angiotensin converting enzyme as a receptor for SARS-CoV virus, or with an angiotensin receptor blocker. We also knew at that time that transplantation was very restricted for these patients. Surgeons didn't want to touch diabetics then because they were thought to have more protoplasm, that their blood vessels were damaged, and that they would not be able to handle the complications of transplantation. So this was the state of the art, really, pretty much until just a couple of years ago. So to pick a year like 2017, what did we know about treating diabetic complications? We knew to control the blood pressure. We knew that it was no longer controversial to control their hyperglycemia. We now has a standard of care, the use of renin-angiotensin blockade, and we know that the drugs that we use also block aldosterone metabolism as part of their activity. And now, of course, transplant surgeons fight with each other to get diabetic patients with end-stage kidney disease for transplantation. But overall, if you look at that, and keeping in mind those figures I showed you of increasing prevalence of diabetes and CKD, this represents, in my opinion, a very poor productivity over a 40-year period of therapeutics to positively impact the effects of diabetes in our patients. So there clearly was a great need to do something about this. And of course, for me, as an investigator, I think of this through the eyes of a pathologist and through the eyes of animal models. So my goal is to identify an animal model that will allow us to identify mechanisms and therapeutics that can interrupt this cycle. So the first question we have to ask in diabetic nephropathy is, what are we trying to model? Now, this reflects back on my role as a diagnostic pathologist, and I will share with you that diabetic nephropathy is a glomerular, tubulointerstitial, and vascular disease. And you see here the lesions in a glomerulus with these characteristic nodules in morphologically advanced diabetic nephropathy, the nodules of Kimmel-Steele-Wilson disease. We see a vascular process in which the muscular arteries of a kidney develop this intimal sclerosing lesion or arteriosclerosis. We see the vascular lesions of diabetes extend to the microvasculature, to the arterioles, where they get filled with this hyaline material, which impedes blood flow and causes a downstream ischemia of glomeruli, but also other aspects of the kidney parenchyma and other organs as well. And that it also involves damage to the tubules and interstitium downstream of the glomerulus that impairs the kidney's excretory functions and contributes to that diminished glomerular filtration rate that I alluded to in the earlier slide. So to understand what we're trying to study in diabetic nephropathy, I'm just going to focus on the glomerulus for the purpose of this talk in order to try and keep within the time limits required. So we all need to have a common understanding of what a glomerulus consists of. What is the architecture of a glomerulus? So I have a normal looking glomerulus over here, and you can see that it's a bundle of capillaries, and you can see that it's a bundle of capillaries supported by a very thin stalk, which we call the mesangium. Now, the mesangium is composed of both cells and matrix, and it supports a clover leaf of capillaries. And the capillaries consist of glomerular endothelial cells with a fenestrated endothelium. They're a highly specialized endothelial cell type, a basement membrane, and sitting on top of the basement membrane are these foot processes. We call them from glomerular epithelial cells or podocytes, and that's the podocyte cell bodies sitting out in the urinary space, and all of this is enclosed by a Bowman's capsule. And so blood comes into the glomerulus through capillary, spreads out through the capillaries, and some of the blood is filtered out into the urinary space where it becomes processed by tubules downstream, and what isn't processed is excreted as urine, while other portions are maintained in the circulation. And so it is often thought of as this as a glomerular filter, this filtration unit that consisting of the endothelium, the basement membrane, and the foot processes that regulates what gets excreted into the urine and what is kept in the circulation. And obviously, if you damage this filter, you're going to have more excretion of proteins and other materials into the urine, and that will manifest as increased proteinuria or albuminuria, a functional marker of glomerular injury. In diabetes, what happens is the mesangium expands and all the parts of the glomerulus effectively get damaged. I already mentioned the hyalinosis of the arterioles. The mesangium gets expanded, the basement membranes get thickened, and podocytes are either lost or reduced in number, and the podocyte foot processes lose their structural integrity, or we say they become effaced. They no longer form these nice discrete units. And so histologically, in a biopsy or an autopsy, this is probably the earliest form of diabetic glomerular injury that we commonly recognize. And that's this expansion of the mesangial regions, typically by matrix, sometimes including a small element of hypercellularity. And so the diffuse mesangial thickening or diffuse mesangial sclerosis is a hallmark of diabetic glomerular injury. But as I showed you earlier, in its most morphologically advanced forms, it's characterized by the formation of these nodules, these Kimmel-Steele-Wilson nodules. And you can see they are a mass of, can be acellular material, matrix, but there also seem to be some disorders of the adjacent capillary loops. They don't look quite like normal capillaries. Some of them look distended, and some of them contain circulating inflammatory cells. And the question is, how do you get to these nodules? Do they come about just by increasing matrix, by deposition of more and more matrix over time, or does something else happen? And I'm going to submit to you something else happens, and what that is suggested again by pathology by the use of a special stain, a silver methenamine stain or a Jones stain, as shown here. So the silver methenamine stain stains glycoproteins black, and that's what you're seeing here. So it stains things like basement membranes, like this Bowman's capsule black. But what it does is it shows varying changes in these glomerular nodules. So in this single glomerulus, I will submit to you, we are able to see changes of chronic injury, acute injury, and repair. The chronic injury is exemplified by this nodule at 6 o'clock, where you can see almost laminations of the silver staining matrix, much like the rings of a tree, which I would submit to you are the sequela of repetitive injuries. The question would be, okay, so what are those injuries? And that's exemplified by the nodule immediately over here at 8 to 9 o'clock, in which you see a loss of the normally compact silver staining matrix, like you might see in a more normal looking mesangium up here. And this dissolution of the matrix is a process we call mesangiolisis, acute injury. And then we can see intermediate stages in these nodules over here, where some of it is compact matrix, but there's also looser areas. And these looser areas contain a number of channels. They're vascular channels, a neovascularization as part of a process of repair. So I would submit that even in a single glomerulus in advanced diabetic nephropathy, we can see acute injury, chronic injury, and not just the possibility, but the actual appearance of reparative mechanisms. Okay. Sometimes the mesangiolisis occurs just as fraying of the interface between the mesangium and the glomerular capillaries, and the capillaries thereby lose their anchoring spots within the mesangium and then blow out under normal pressures and flows to create these microaneurysms like you see here. So mesangiolisis and diabetic nephropathy involves fraying of the mesangium, lamination of the matrix. It's implicated in the formation of mesangial nodules and can result in the formation of capillary microaneurysms. All right, so now we've talked a little bit about the mesangium. Let's go on and talk about the podocytes. We really want to pay attention to them and ask the question, do they have a role in diabetic nephropathy? So let's talk a little bit about podocytes and look at them in a little more detail. So on top over here, we have a scanning electron micrograph, which shows a podocyte body with these processes that extend out from it. And these processes break up into these very small processes, which then become the podocyte foot processes, which sit on the basement membrane. And that's depicted in a two-dimensional transmission electron micrograph, where you see this podocyte cell body sitting in the urinary space, putting down foot processes on the capillary basement membranes, forming part of that filtration unit. So one of the things you can see from the scanning electron micrograph is that this is a very complex supporting architecture. In many ways, you might even imagine they look like the buttresses that support a Gothic cathedral. So they provide important support to the capillary network, and they counteract capillary wall distension and hydrostatic forces to maintain capillary structure, such that if you lose podocytes, you lose that support and you allow capillaries to blow out to form microaneurysms. Other important facts about podocytes: they are terminally differentiated with very limited replicative capacity. In fact, so limited that people have long said that podocytes, once lost, cannot be restored. And that this is a process that leads to irreversible renal failure when they are lost in sufficient numbers. Podocytes are important for synthesizing portions of the glomerular basement membrane, and as I've indicated, they form with the glomerular basement membrane and endothelium a barrier function, a filtration barrier to prevent abnormal proteinuria. One thing not mentioned in the slide is that podocytes are also synthetic cells, and they make important proteins and growth factors such as vascular endothelial growth factor that may be trophic to other cell types and engage in crosstalk with other cells in the glomerulus, such as the endothelium and mesangium, and that if you lose these podocytes or their trophic factor secretory functions, that too can cause a kind of injury due to the loss of this trophic function. We know from studies by others in clinical settings that low podocyte number leads to poor outcomes in diabetic kidney disease, and that's true whether patients have their diabetes from type 2 diabetes, increased insulin resistance, or type 1 diabetes due to pancreatic islet destruction. This is just one paper looking at type 2 diabetics in the Pima Indian population. So what do we know about podocyte loss in diabetic nephropathy? What we know is that podocyte loss is a feature of diabetic glomerulosclerosis based on studies like the one I just briefly alluded to. We know that podocytes have a limited capacity for replication, and again, that has been used to suggest that diabetic nephropathy is a linear and progressive disorder that does not allow for regression, and that has certainly been basic understanding for many years. That podocyte loss or loss of function can lead to mesangiolisis. I'm going to show you some studies from Acuna Ichikawa in the following slides, but also if you interfere with the trophic function, such as the secretion of VEGF, either by knocking out VEGF in animal models, as is done by the laboratory of Sue Quaggan, or look at the impact of therapeutics that interfere with VEGF, such as Avastin or bevacizumab, that those effects, whether loss of the actual expression or interfering with the function of the secreted molecules, can also lead to mesangiolisis. And I have showed you at least a scenario that mesangiolisis can lead over a series of episodes of repair, injury and repair can lead to nodular sclerosis. So here are the studies from Ichikawa that I alluded to. This is really an intriguing study. So what they did is they took mice and genetically put a human diphtheria toxin receptor strictly on the podocytes of these mice. Mice don't normally have a receptor for the diphtheria toxin, and so if you give them the toxin, it doesn't do anything to them. The podocytes are marked by stains, a nuclear stain for WT1, which stains darkly here, and you see the complement of podocytes in this mouse, Glomerulus. If you then give diphtheria toxin to these mice, you kill the podocytes, but nothing else. And so here's an example of a mouse with the engineered diphtheria toxin receptor that has received the toxin and lost all of its podocytes. Now, Ichikawa and his colleagues were using this model to study the effects of podocyte loss, titrated podocyte loss, on the development of glomerular sclerosis. But I had a different interest in their studies, and so I asked Agnes Fogo, the pathologist who worked with the Iacuni, if we could have their tissue blocks from this study, and if we could stain them with our silver methenamine stains. And Agnes kindly provided these blocks, and what we were able to show is that by killing the podocytes in these mice, you could create mesangiolisis. We see the loss of that compact mesangial matrix, mesangial structures, and we see the development of these capillary microaneurysms in the mouse glomeruli, sort of recapitulating those scenarios that we saw in the humans. So, taking all of that together, if podocyte loss in diabetic nephropathy, if podocyte loss is a feature of diabetic glomerular sclerosis, and if podocytes have a limited capacity for replication, how can we account for the possibility even of regression of diabetic glomerular sclerosis? And so I suppose the first question we have to ask is, can we actually get regression of diabetic glomerular sclerosis? Is diabetic nephropathy reversible? So here I want to show you the results of an absolutely extraordinary study published in 1998 in the New England Journal of Medicine. This comes from Minnesota, where they had a long-standing study of type 1 diabetics who had agreed to undergo protocol renal biopsies. Now, a subpopulation of those patients underwent a pancreas transplantation-only procedure to try and overcome the loss of the pancreatic islets that caused the type 1 diabetes. So following these patients over time, they had a baseline biopsy establishing the presence of diabetic glomerular sclerosis, including these big nodules, and protocol biopsies obtained 5 years after pancreatic transplantation showed no improvement whatsoever in the kidney structures. A very disappointing finding that nonetheless was published in the New England Journal in 1993. But at 10 years following pancreas transplantation, a repeat protocol biopsy showed marked regression in all of the 8 patients entered into the study. 10 years after pancreas-only transplantation showed reversibility of established diabetic nephropathy. So the question now is, wow, proof of principle established. How do we figure out what enables the reversibility of diabetic glomerular sclerosis? And again, for me as a pathologist, my natural inclination is to think about animal models that will help us recapitulate this process and enable us to study it. Now, I will tell you that over the years, I have, and I, along with my laboratory partner in crime, Kelly Hutkins, have looked at well over 40 models of diabetic nephropathy. That's probably a tremendous underestimate. We have looked at ones where we vary the their lipid status. We have looked at various genetic mutations, at knock-ins, at knockouts. For a number of years, we were the reference laboratory to look at diabetic injuries in mouse models of the Jackson laboratories. I'm not going to tell you about all of those models. I'm going to tell you about just one. And this is one that we have characterized in our laboratory. It's the BTBR-OBOB mouse model. And this is why I've chosen to talk about it. It is a model of podocyte loss and restoration. In fact, it's the only animal model of podocyte loss and restoration in diabetes. It is a model in which the loss of podocytes can be linked to mesangiolisis. It can be linked to the accumulation of reactive oxygen species in podocytes, which we think may be the initiating injury causing podocyte loss, but which we won't have time to go over today. And it's a useful model for testing efficacy of therapeutics. And these are some of the therapeutics I hope to discuss if I can keep talking at this rate of speed and keep to my time. We'll talk about the inhibition of renin-angiotensin-aldosterone system drugs, reversibility with leptin, the newer drugs that have come since 2017 for the treatment of diabetes, Sglt2 inhibitors, and endothelin receptor blockers. And we won't again have time to talk about reducing reactive oxygen species. So to introduce the mouse, the BTBR strain is a strain, it's a type of mouse, and it's had the OBOB or leptin deficiency mutation introduced. So if you look at these mice physically, you can see on the top the BTBR wild type and heterozygote, and they are of normal sized mouse. The heterozygote has a skin coloration which comes along with the OBOB mutation. But at the bottom, you see something very different in the homozygote BTBR-OBOB mouse. So with leptin deficiency, mice develop really a single purpose in life. It's actually kind of true for the rare humans who also have leptin deficiency, and that is incessant hunger. These mice live to eat. They eat, and they become fat, very fat, as you can see here. They become so fat that then so interested in eating, they lose interest in everything else, including sex. So it's very hard to breed up BTBR, OBOB mouse colonies, in part for that reason. Functionally, the BTBR wild types and heterozygotes have normal glucose, lipids. They don't have functional evidence of renal dysfunction. But with the OBOB leptin deficiency mutation, they become true type 2 diabetics. They're obese, they have markedly elevated glucose, their lipids go up, and they develop kidney disease as manifest by proteinuria. And here you see the actual measures of kidney disease over here circled. You can see the markedly elevated protein excretion of BTBR OBOB mice compared to mice with the OBOB mutation in other strains in which they get mildly increased proteinuria, but not very much, and don't get the structural lesions of diabetic nephropathy. These mice do get structural lesions of diabetic nephropathy, so you can see at 14 weeks the marked mesangial expansion that these mice can develop. This is also much more rapid than most mouse models, which normally take 6 months or more to develop characteristic diabetic lesions. The fact that these mice can develop in a somewhat accelerated time frame makes them a good choice for studying therapeutic interventions because you reduce the time needed to develop lesions and assess the efficacy of any interventions you may employ. The other critical thing about the glomeruli in these mice is they develop mesangiolisis, and you can see that here. And so for all the reasons that we've discussed up to now, this makes them a very attractive model to think about how they could be studied for the regression of diabetic nephropathy. So reversing diabetic nephropathy, again, this is how we do it. It's going to sound like we had a straightforward plan for accomplishing this. We didn't. It was actually a very complicated roundabout plan, but in the interest of time, we'll give the straightforward, uncomplicated version. And what we did is we essentially gave them back the leptin. We did it by implanting mini pumps into the backs of the mice. These pumps allow the steady release of leptin into the circulation. And when the mice get the leptin back, it changes their outlook on life. They stop eating. And it's good to know that in mice, just as in people, if you stop eating, you lose weight. And they lose a significant amount of weight. And you can see that in this line indicating the mice who received the leptin therapy. And if you lose weight, your diabetes changes. You're no longer insulin resistant. So because of insulin resistance, BTBR OB mice have to have very high levels of insulin to maintain any kind of glucose homeostasis. But with the loss of that insulin, with the loss of weight, the insulin resistance resolves and their insulin levels return to baseline. Importantly, as you then lose your insulin resistance, you lose weight, what makes the nutritionist happy and what we tell type 2 diabetic patients, if you lose weight, your glucose will get under better control, and that's as true in the mice as it is true for most individuals with type 2 diabetes. So they have really improved their diabetes. Now let's get back to diabetic nephropathy. I showed you this slide earlier. It's a landmark slide. It proves that in humans, the species we do care about the most, diabetic nephropathy is reversible. And what I would say to those of you who are listening, if you could remember 2 slides from this entire talk, this is one of the slides I would like you to remember. Diabetic nephropathy is reversible. Now, in the leptin-treated OB mouse, this is what happens. And this is the second slide that I would like you to remember if you were to only remember 2 slides from the entire talk. Here is the BTBR wild-type mouse with a normal-looking glomerulus. Here's the OB mouse with marked mesangial changes and other changes of diabetic nephropathy. If you allow the mouse to go on for another 8 weeks, these changes are progressive. There's mesangiolisis. There's marked mesangial expansion. And if you take that 20-week BTBR OB mouse and give it 8 weeks of treatment with leptin, you can see the reversion to normal of the glomeruli, so that diabetic nephropathy is reversible just as it is in those patients receiving a pancreatic transplant. And we can look at it through a variety of ways. This is just one picture looking at one of the matrix components of diabetic mesangium, here collagen 4, showing that it increases in the diabetic mice. You lose it as you get regression of the nephropathy. And you look at a variety of matrix molecules and show similar findings. Functionally, it's not a surprise. If you make the structure better and you get them to return to normal, you should get improvement in all the functional parameters, and you do. So here you can look at proteinuria, BTBR wild type without significant proteinuria, marked proteinuria in the mice at 20 weeks. And again, that is regressed when the structural lesions regress with leptin therapy. Back to the podocytes. So remember, the proteinuria comes about through damage to the glomerular filter. The filter is composed of podocytes, basement membrane changes, and the endothelium. We're going to focus on the podocytes. So here you look at the podocytes in a BTBR wild-type mouse stained with that same WT1 marker that was used in the studies by Ichikawa. So here's a normal complement of podocytes, markedly diminished with diabetic nephropathy in the OB mouse, just as they are in humans, just as they are. But with treatment, the podocytes come back. So there's not an absolute limitation on the ability for podocytes to be restored. And this is one of the early demonstrations for the possibility of regression. And obviously it is occurring in the setting of regression of diabetic nephropathy. And this is just a graph to show those things. Whoops. The graph showing that here's the podocytes are lost early in the course of diabetic nephropathy in BTBR OB mice. They're lost as early as 8 weeks and probably earlier. 8 weeks is just the earliest time point that we've really measured in detail. They stay lost at 20 weeks compared to the wild type, but that they're restored when you give the leptin therapy for a period of 8 weeks. So thinking about this on what I call the podocentric view of diabetic nephropathy, how would you replace these lost podocytes? That's our next question. Can it be from proliferation of existing podocytes? And I think the literature is pretty strong that podocytes don't reproduce from other podocytes, that they are terminally differentiated, leading to this concept that you can't get restoration of podocytes. But we live in an age of regenerative medicine, so maybe there's possibility of regeneration from a local progenitor source, or maybe from a non-local progenitor source. And people have looked at tried to prove scenario number 3 without any significant success. So that leaves us with Possibility number two, that there is a local progenitor source for these restored podocytes. And the answer came, of course, from our pathology studies, again, using the WT1 staining. So if you look at the wild type, here's our normal complement of podocytes, lost with BTBR-OB, restored with leptin, but those cells lining that Bowman's capsule that enclosed the urinary space that weren't staining with WT1 in these other scenarios now are lighting up with WT1. They're acquiring podocyte-like features. We believe those are at least one potential site or one location of progenitor cells that can lead to podocyte restoration. So we did a broad study. We looked at BTBR OB mice treated with leptin, but also treated with the standard therapies that have been used for 40 years for treatment of diabetic complications to see why don't patients get significantly better. It delays the progression of their kidney disease, but not reversal. And we looked at this issue of podocyte restorations. So here we have the BTBR wild type with its normal complement of podocytes, the loss of podocytes in the BTBR OB mice, no change with a control group getting only blood pressure control with hydralazine, no change with losartan, that's the angiotensin receptor blocker, no change with the treatment with enalapril, the angiotensin converting enzyme inhibitor, but marked change with leptin. So we speculated that one of the reasons that current therapies, at least through 2017, 2018, fail to improve the actual trajectory of diabetic nephropathy was by their failure to enable podocyte restoration. Now, we also looked at the ultrastructural features and said, well, are these really functional looking podocytes? We believe they were because we saw the reduction in proteinuria, and so basically a return to a normal filtration unit, but electron microscopy just demonstrated between the wild type OB and those treated with leptin that podocyte foot processes seen here at high power in the wild type mice lost or effaced in the OB mice that are untreated become restored when you retreat with leptin. So there's an ultrastructural structural integrity that's restored with these newly formed podocytes as well. So basically that gives rise to a thought that the glomerular tuft has in this Bowman's capsule a source of renal progenitor cells that can be mobilized under certain circumstances to restore lost podocytes and enable regression of diabetic nephropathy. Okay, so this is just to anticipate some questions. We have done the most obvious thing you would do, or among the obvious things one would do today, is do a transcriptomic analysis through the course of regression, beginning early through late, to see if we could establish pathways. And we have data that gives some clues as to important pathways that are activated in the course of the trajectory of regression. But we really don't have time to go into that and in this talk, and also our analyses are still pretty rudimentary. But we would welcome people who are interested in this sort of thing to join us and see if they can help us parse out some of the important pathways that are activated in the course of regression. And we'll still come back to some transcriptomic analyses if there's time at the very end of this talk. So now I want to get us into what was the goal that we had basically when I was still a house officer in internal medicine. And how do we have better treatments, better ways of taking care of our patients with diabetic complications in general and diabetic nephropathy in particular? And so I'm going to first talk about a class of drugs, Sglt2 inhibitors, and in this case, a very specific one, empagliflozin. So Sglt2 inhibitors have a principal mechanism of action where they act on the sodium glucose cotransporter 2, which is located in the proximal tubules of kidneys. And what they do is they block SGLT2 normally works to bring sodium and glucose that's present in the filtrate that's been filtered across the glomerular filter into the tubules and reabsorbs it back in to regulate the amount of sodium and glucose we have in the circulation. But if you block the ability of the proximal tubule to absorb sodium or glucose, you get a number of effects. For one is you're going to get a lot more glucose in the urine. And by having the glucose in the urine, it's not absorbed, so there's less glucose in the circulation, and you achieve a measure of glycemic control in that respect, and to some degree it contributes to weight loss. Similarly, if you don't absorb all the sodium that's filtered in the urine, you're going to have some hemodynamic changes that result in lowered blood pressure and lower intravascular volume. In taking advantage of that physiologic element, the drugs were used initially in broad clinical trials to treat patients with heart failure to see if they could reduce the volume overload and hemodynamic aspects of heart failure. And they were tremendously successful in those trials, but a secondary outcome was to demonstrate particular improvement in renal function in those patients who were diabetic and had heart failure and were in those trials. So that led to the publication of this absolutely landmark study 2 years ago in the New England Journal. And this looked at another member of the class of Sglt2 inhibitors, canagliflozin, in patients with type 2 diabetes and nephropathy. And what this study showed is that no matter what renal parameter you looked at, whether it be GFR, a measure of renal excretory function, urinary albumin to creatinine ratios, a measure of albumin excretion, or any composite by any measure whatsoever, patients did better if they were treated with an Sglt2 inhibitor. And that extended to other features like death from cardiovascular causes or death from any cause or prevention of kidney disease. All of those really benefit from Sglt2 inhibition. So the question is, why? Those are functional improvements, but are there any other improvements that might predict a long-term benefit? And these are short-term studies. So we started a clinical trial of our own, and it had a very restricted class of individuals that could be entered into the trial. You had to be a fat BTBR OB mouse or a controlled skinny BTBR wild-type mouse. We treated them, patients either were untreated, treated with leptin, or treated with an Sglt2 inhibitor. And when we see there's a bunch of clinical data here, but I've bolded the critical ones, we did achieve better glucose control with the treatment with the Sglt2 inhibitor. We had a trend, but not statistically significant improvement in serum creatinine after 6 weeks of treatment. But we had noticeably improved reduction in proteinuria and urinary albumin excretion with the Sglt2 inhibitor treatment. Along with that, we had tremendous improvement in the structural features. So this is something that was not known from the treatment of patients or clinical features, what structurally was happening, because none of these patients had renal biopsies. So we see a reduction in the mesangial matrix and a reduction in the mesangiolisis in the graphs. Each one of the points represents an individual mouse treated for 8 weeks. We see, again, following our studies of podocyte density, now you're used to seeing them, wild-type mouse, normal complement, reduction in podocytes with the OBOB, restoration with leptin, and similarly seeing restoration with the Sglt2 inhibitors. This is something that was not known and unanticipated benefit of treatment with these drugs. And similarly, using a transmission electron microscopy, we can show restoration of podocyte foot process integrity with this treatment. So with the Sglt2 inhibitors, we can include directly or indirectly, empagliflozin can promote structural improvement of diabetic nephropathy, and it's now been shown in several mouse models. Empagliflozin may enable regression of established diabetic nephropathy, that podocyte restoration is achieved as the regression proceeds, and that the mechanisms of regression may be associated with reduction of reactive oxygen species in specific cell types, including podocytes, but again, not time to show that. I want you to go to a second vignette of endothelin receptor antagonist therapy. And endothelin is a systemic hormone that acts via either an endothelin receptor A or B. When it acts through endothelin receptor A, which is located on multiple cell types. Among its effects are a hemodynamic effect. It can be a vasoconstrictor, and so antagonism of the endothelin receptor is thought to allow vascular relaxation, greater blood flow, and there are a number of downstream consequences of those hemodynamic changes that may result in improved renal structures as a result of this therapy, particularly if there is associated hypertension, as often occurs in type 2 diabetes. So knowing this physiology, there was a relatively small study called RADAR published about 6 or 7 years ago, which used the endothelin receptor antagonist atrasentan, and it was found that it improved protein excretion in patients with diabetic nephropathy, patients with type 2 diabetes. This led to a much bigger trial called the Sonar Trial, published almost exactly the same time as the Credence trial. This one was published in the Lancet, and it too showed a tremendous benefit of endothelin receptor blockade on renal excretory function and to some degree on proteinuria. So two major advances in renal therapy, all just in the last couple of years. So we had also been aware of the RADAR study and did a controlled clinical trial of atrasentan with the angiotensin receptor blocker losartan in our BTBR mice. And that's because in the clinical trials, the atrasentan was added to standard therapy, which involved either treatment with an angiotensin receptor blocker or an angiotensin converting enzyme inhibitor. So in this study, again, we find the BTBR wild type mouse, no change when it's treated with atrasentan. The glomerular injury of the OBOB mouse, which improves with atrasentan and improves maybe even a little bit more with atrasentan plus losartan. And you can see that visually here. Similarly, if we look at podocytes again, our standard mode of operation, we show the wild type normal complement of podocytes, no change with atrasentan, loss of podocytes with the OBOB mutation, and some regain of podocytes with the atrasentan, and even more so with the atrasentan plus losartan. And again, we show that the restored podocytes demonstrate structural integrity with retention of podocyte foot processes, and that work was all published in the past year. We also see that with the structural changes, that the functional changes of proteinuria also occur with the treatment with atrasentan and losartan, and that there's improvement in glomerular excretory function as well. So we can conclude from the atrasentan studies that directly or indirectly, atrasentan can promote structural improvement of diabetic nephropathy in a mouse model, that it may enable regression of established diabetic nephropathy, and that podocyte restoration is achieved as regression proceeds. Now, I'm just going to add a small but complicated study as a finale, and that's to bring in the transcriptomics data that I mentioned earlier, but in a different setting. So we did similar glomerular isolations and transcriptomics in the atrasentan-treated mice whose pathology I just showed you. We also know that the patients from that RADAR trial had urine, serum, and plasma retained, and a variety of omics studies were performed on those biological samples, including studies using miRNA to reverse assess mRNA expression in cells in these patients. Those studies were all performed at the University of Michigan as part of the BEAT DKD consortium. And what was then done was we separated out the patients in the RADAR study who were responders and non-responders, and particularly wanted to study the responders and compare their changes in mRNA expression, or transcriptomic profiles, with those of the regressing BTBR OBOB mice treated in a similar way with atrasentan and losartan. And what we learned was, if you just took the OBOB mice compared to the wild type mice, that there are 1,353 genes that are changed, at least using as assessed using the Affymetrix chips that were available to us at that time. The atrasentan protocol reversed those changes in 1,068 of those genes. And that's shown broadly in this graph where each dot represents an individual gene. And on this side of this vertical line are all the genes that were upregulated in the OBOB mice. And on this side of that line are all the genes that were downregulated. And in this portion of the graph are those portions of the upregulated genes that were reversed with atrasentan. And this part of the graph, those part of the downregulated genes that were reversed by atrasentan. and the remaining squares contain the genes that were unaffected by the atrasentan protocol. So as an example, here's one gene, a gene that's I'm having trouble reading it, but it's basically a gene that's it codes for a phospholipase of still unknown specificity. But this gene is markedly upregulated in the OB mice, downregulated with atrasentan treatment. This gene is a protein, codes for a gene that's related to the frizzled-related protein family, so it binds to Wnt receptors, the Wnt proteins, and modulates Wnt protein signaling. And this gene is downregulated in the OB mouse, upregulated with atrasentan. And again, you can see where these genes plot out on the big graph. Okay, so there are a number of genes that are upregulated and downregulated. When you put them into pathway analysis, you can find that there are a number of pathways that are upregulated, of course, or downregulated or modified in the OB mouse. But what was striking was the pathways that were modulated in the OB mouse were the same ones that characterize the responders versus the non-responders in the patients in the RADAR trial. So it suggests that our model can be used to test various therapeutics in preclinical trials and actually identify pathways that lead to responses. And this is starting to attract some significant attention, and it's a very exciting development for us. So with that, I'm going to conclude with these thoughts with you. Diabetic nephropathy is a reversible injury, but only under certain conditions at present. And these include if you're a type 1 diabetic with a 10-year pancreas transplant, or if you're a BTBR OB mouse treated with leptin, possibly with some of these new drugs or with bariatric surgery, which we haven't discussed. Podocyte regeneration is key to this process. Podocyte regeneration likely comes from parietal epithelial cell progenitors, but that is not a proven scenario, just suggested. There is phenotypic plasticity between podocytes and PECs in human diabetics, again, the species we care about the most. And a lovely study showing this was done by Nicole Andine when she was a resident and fellow in our program here in Seattle. And some current therapies for diabetic nephropathy may enable podocyte replacement and long-term contribute to disease regression. So with my last slide, I'll thank some key members of the lab. Currently, Kelly Hutkins and James Lee do all of the work in the lab, and Kelly has been with me from the start. She was the first person to work in my lab going back to 1988, and we're still working together as a team, and all of the good work presented here was either done by her or led by her, or supervised by her. Can't be thanked enough. Our collaborators for the transcriptomics come from the University of Michigan. Within the U, I didn't talk about our ROS studies or our vascular studies with Peter Rabinovich or Jenny Cantor. Renee LeBoeuf was early on a key leader of helping us understand animal models of diabetes. And Alan Addy and Mark Keller get credit for putting the OB mutation into the BTBR mouse. We've received funding from a number of corporate entities, from the BTKD Consortium, and obviously from all of you through your contributions to the NIH. And so I thank you very much for your attention and interest.
Thank you, Charlie. That's a wonderful story and wonderful progress. So I don't see any questions in the chat, but I have questions. But are there questions out there? And please speak up. Anyone?
I have a question. This is Erica.
Yeah, Erica.
I was really intrigued by, thank you for your talk. It's just amazing and so educational for me. I was really intrigued by those results with podocytes coming back. You know, as you said, they don't regenerate yet. You know, and 5 years after pancreas transplant, they're not there, and suddenly at 10 years they are there. And I was wondering, why is this taking so long, and what is initiating it? Do you know much about the mechanism? It's just so interesting that they do come back, and they're like new podocytes. I guess they're not old ones that regenerated. Can you say more about that?
Probably not a whole lot beyond that, but I'll try. So thank you for your compliment and interest. So we like the mouse model in part because it's fast enough to do some of these things and explore it. As you say, the human is 10 years and we don't have sequential biopsies, so we can't get that information. And the biopsies they had, they've been used for a lot of studies, they've largely been exhausted. So the idea is that there is probably podocyte regeneration, but we don't really know the mechanisms why. It's a very difficult problem. It obviously elicits a lot of interest. But the research, again, these are people, and why we we need the animal models. So I wish I could be more informative than that, but I can't.
Well, there's a question in the chat. Impressive work, so important to have a model of diabetic kidney disease replicating so many aspects of human. 2 questions about drugs. Any new insights into mechanisms of kidney protection of Sglt2 through BTBR? And have you investigated GLP-1RA with their anti-obesity and kidney effects?
Right. So we have not done the GLP-1 receptor antagonists, which we would have liked to have done, but have not. That takes care of the other. For the protective mechanisms, we have engaged. So the empagliflozin treated mice have had their glomeruli isolated. They have been made available for transcriptomic analysis with our collaborators in Michigan. We have not done the actual transcriptomics ourselves. They were set to go just before COVID hit the world, and everybody's laboratories were severely restricted in their activities. And while some of this you could say could be done from home and all, lots of things get done, and that group has basically put this on the back burner. We're hoping to learn from comparisons with the wild types who are also treated with these drugs, some information that may have a protective effect, but we don't have that at hand as yet.
Charlie, question from Mark Weiner. And are there urine or serum biomarkers of podocyte status?
There are actually. I'm sort of the wrong person to answer that, because my colleague Behzad Najafian really does substantial, wonderful work and has looked at things like exosomes to do that. People have looked first just to see whether you can get podocytes and be able to culture them out of urine. You can. We're trying to get a quantitative sense of how many you excrete, and could that serve as a biomarker? And that's been difficult to do. There are a number of new exciting studies that I am only peripherally involved in because I'm part of a large consortium called the Kidney Precision Medicine Project that says maybe we actually can do some transcriptomics on those podocytes captured from the urine in these patients with diabetes that may allow us to use them as markers of degrees of injury. But more simple markers that we'd like to employ immediately in the clinical laboratory, I don't believe exist, or we would be trying to employ them in the clinical laboratory. So again, not the most satisfying answer, Mark, but I think that's where we are.
Charlie, a question from Peter Rubinovich. Great talk. Can you speculate on the common features of Sglt2 and the atrasentan models that may help explain podocyte regeneration? Right.
So I was careful to say that I think the benefits could be direct or indirect. And a guess, my hypothesis would be that the effects are indirect and may have a lot to do with hemodynamics. Both of those drugs affect intravascular volumes and hemodynamics, particularly at the level of glomerular arterioles. And that would be a leading thought. But the specifics of how they do that, I don't yet know. And I see Behzad has a question, but he may also want to weigh in on urinary markers or biomarkers of podocyte injury in addition to his question. Do you want to do that, Behzad? Can you
yeah, Charlie, thank you. Well, podocyte loss through quantification of the number of podocytes in the urine can be evaluated. It's not a routine or commonly available test. We do that for some clinical trials. It is a challenging test currently to be done, as Charlie mentioned. Performing transcriptomics and podocytes that you can capture from the urine of patients is a new field that there are different investigators doing that, as well as a study of the exosomes. But there is progress in the methodology. Each of these are faced with different challenges from practical point of view, especially if thinking of bringing them into clinical field. But there has been progress made, and hopefully, I mean, in the future, we can think of non-invasive biomarkers of podocyte loss.
Good. So then I see in the chat you had thank you for adding all of that. You are the expert. Thank you for asking. Do I think podocyte injury is an early driver of diabetic nephropathy? And do we need podocyte restoration for reversal of diabetic nephropathy? The second part is, I think, easy for me. I really do believe you need some kind of podocyte restoration to reverse diabetic nephropathy, or at least diabetic glomerular injury. As I alluded to at the very beginning, but otherwise sidestepped, there's a lot of diabetic injuries, as you know, that may be limited to the vasculature or tubulointerstitium, and for those, podocytes may not be a relevant target or concern. But for reversal of glomerular injury or morphologically advanced glomerular injury, yes, I do believe we need podocyte restoration. Do I think of podocyte injury as an early driver of diabetic nephropathy? I do. I don't do it with complete certainty, but I think those processes of both that lead to mesangial expansion and things clearly are driven by metabolic effects on the mesangium and endothelium itself. But I think we all see, even in our clinical biopsies, the occasional glomerulus in a biopsy of a diabetic patient without much morphologically advanced injury that shows the segments with mesangiolisis and stuff. And I think those early events, before there's been the buildup, before you've developed a nodular sclerosis or even significant mesangial sclerosis, are still mediated by podocyte injuries. So not necessarily even podocyte loss, but something that's interfering with their production of critical trophic factors. Well, yes, I think so.
Let me ask a really basic question, probably pretty stupid, but where does glucose figure in?
It's tough because we see people with really excellent glucose control still having diabetic complications. They can have all of them. There's no question that having good glucose control reduces your risk of complications and maybe even the severity in many patients, but not all. So it can't be the sole explanation. It's clearly important. It drives a whole series of downstream metabolic changes, both in individual organs that then have other effects, like in adipose tissue. It drives certain inflammatory changes. It drives the formation of advanced glycation end products and a variety of structures, but it doesn't do the same for everyone. And so maybe this is a complex interaction of glucose, underlying genetic predispositions. I know those are easy answers to throw out for uncertainty, but they may be real because it's unclear how it is that some folks with good glucose control don't really get better. And although I realize we're at the end of the time, if he were still on, one of the great leaders in these investigations, and the leader of the recent KDIGO discussion of treatment of diabetes is Ian DeBoer, and I saw from the chat that he was on. He might still be there and might want to weigh in on that. And again, he is the world's expert.
We can
may not be here. May not be here.
Okay.
But I mean,
yeah. Great. Okay. Well, Charlie, That completes a wonderful hour. And thank you for sharing your career with us. This is a marvelous story. And for attendees who need CME, Brooke Emmerich is going to send out in the next day or two a email that contains information about how to get CME credit. So with that, let us close a great hour, Charlie. Thanks again.
Thank you, Larry. Thank you all.